In this chapter, we'll discuss how to classify organisms. So why is classification helpful? Classification is primarily for convenience. It's a human construct that scientists have developed. Grouping organisms allows scientists to organize information and characteristics rather than listing random facts. The science of classifying organisms is called taxonomy. So here's an example of why taxonomy and classification would be important. Imagine going to a grocery store and trying to find the lettuce if nothing was grouped or organized. So by classifying all of the vegetables into the produce section, you can know exactly where you can find that lettuce. How did Linnaeus change taxonomy? Aristotle was the first to classify organisms. He classified into 6 groups based on appearance. As you might imagine, there were many flaws in this method because some of the organisms seem to overlap groups or not fit well into any group. So 6 groups is pretty small for the number of organisms that they knew existed even then. Swedish botanist Carolus Linnaeus created a better way to classify living things. He used a hierarchy of groups to show similarities between organisms. So we'll look at the hierarchy here. It starts with a kingdom, which is the largest category that he used. And below that is phylum. So this would contain fewer types of organisms than a kingdom. Followed by class, order, family, genus, and then species. And a good way to remember this is King Philip came over for great spaghetti, if you need to remember those in order. And so they're organized here from the most general category, which would be kingdom, to the most specific, which would be species. What is binomial nomenclature? So although this system is still used today, it was not Linnaeus's most important contribution. His most important contribution was binomial nomenclature, which is a little redundant here, but bi meaning to, nomial names, to name naming system. Previously, common names were used for organisms, and this was problematic because people used different common names for the same organism. So for example, this creature here is known, depending on who you're talking to, as a pill bug, a potato bug, a roly-poly, or a wood bug, and it also has several other common names. So it would be difficult to tell that you're discussing the same organism if you were using different common names for it. In binomial nomenclature, each species is given 2 Latin names, genus and species. So 2 Latin names for a particular organism, and the first word is the genus and the second word is the species. Latin was proposed because it was a dead language, so it would be consistent for the ages. So the scientific name for this insect is Armadillidium vulgare. And anytime a scientist was using, or anyone was discussing this organism, they would use this particular scientific name. They might still use a common name, but they would know they're discussing the same organism because they're using that binomial nomenclature scientific name. So in summary, classification is helpful for organizing information into groups. Although others unsuccessfully tried to classify organisms, Linnaeus used a hierarchy and binomial nomenclature, which are still used today. So how are organisms grouped? We discussed the grouping system, but how are they grouped specifically? Organisms in the same species can interbreed. So any organisms that can breed with each other are going to be in the same species. And we usually go a little bit further to say that they have to be able to interbreed and produce viable offspring, or offspring that continue to breed as well. Larger groups are harder to determine because they are based on similarities and differences between species. For example, ticks and spiders. So here's a tick and here's a spider. Both have 8 legs, but ticks are parasitic and they don't build webs. Should they be in the same category? So they have some similarities, but they have some differences, and scientists need to decide if they could be or should be in the same category. Studying the genes of these organisms does give some greater explanation and ability for scientists to classify things a little bit more consistently based on how similar their DNA is. Why do classification systems change? So originally there were only 2 kingdoms of organisms, plants and animals. As technology changed and scientists discovered more organisms, additional categories were formed. And that continues today. We continue to find new species, and they need to either be classified into one of the categories that we've already come up with, or they need to start a new category of their own. So this slime mold might originally have been categorized as a plant, but it has different cell walls and it has no chloroplasts. As you can see, it's not green. It's now categorized as a protist. Genetic analysis provided even more information on the similarities between species. And currently, organisms are now grouped into 6 kingdoms within 3 domains. So a domain is a larger category than a kingdom, and one of the more recent additions in the classification system. So here are the 6 kingdoms: eubacteria, archaebacteria, protista, fungi, plantae, and animalia. So we'll discuss why this protista kingdom is in quotation marks in a minute. But these are classified into 3 domains then. Eubacteria are in the domain bacteria, archaebacteria are in the domain archaea, and then the other 4 kingdoms are in the domain eukarya because they are eukaryotic and contain eukaryotic cells. These 2 are prokaryotic, but they're different enough that they have their own domains. So what are the characteristics of the 6 kingdoms? Bacteria here, we've got eubacteria, archaebacteria, and then our 4 eukaryotic kingdoms, and we'll go through each one. So within the kingdom eubacteria, these are the most abundant organisms on Earth. They're unicellular and, as I said, prokaryotic, and they are often known to cause disease or are decomposers. So these would be the bacteria that you think of the most frequently, that you would encounter infecting your body or breaking down a dead log or leaves, something like that. Kingdom archaebacteria, on the other hand, are unicellular and prokaryotic as well, but they live in extreme environments like undersea vents, acidic and salty environments, and they survive where other organisms are not able to survive. So that makes them drastically different than the eubacteria, which is why they are in their own kingdom and own domain. So this hot spring in Yellowstone National Park gets its colorful decoration here on the edge, the yellows and oranges, from a variety of Archaebacteria species. And that is a thermal spring, and so it's extremely warm and therefore difficult for other organisms to survive in. Domain eukarya all have eukaryotic cells with a true nucleus and membrane-bound organelles. The domain eukarya includes organisms with the largest body mass and size. So these are the ones that you're going to be the most familiar with. And we're going to start with the kingdom Protista. They are unicellular, they can be multicellular, or they can be colonial. So lots of different types of organisms in this kingdom. They can be heterotrophic or autotrophic. And because there's so much variety in this kingdom, scientists no longer accept it as a kingdom, which is why it's got the quotation marks here. Because as they've started studying the genetics of these organisms, they realize that they don't show any evolutionary relationships in the genes, and therefore they are getting lumped into this category but don't necessarily belong here. They belong in a different category. And separated based on characteristics. But for now, organisms in this category have a variety of sizes, shapes, nutrition, and movement. So we've got very large algae in this category. We have microscopic organisms in this category. We have heterotrophic organisms. We have organisms that are able to move, are unable to move. And so really a great variety within this kingdom. Kingdom fungi are mostly multicellular. There are a few unicellular ones, like yeast is a unicellular fungus. They're heterotrophic decomposers. In other words, they break down matter. And fungi were originally classified as plants. But other than often being anchored in the ground, and not even always being anchored in the ground, they're actually quite different from plants. So they do not have the same type of cell walls, and they don't make their own food. They digest their food externally and then absorb the material in. And so really pretty different than plants, other than maybe the way that they appear growing from the ground. Kingdom Plantae is autotrophic. They perform photosynthesis to make their own food. They are stationary. And they range in size from tiny mosses to giant sequoias. Kingdom Animalia are heterotrophic. They do not make their own food. They are multicellular, although there can be a variety of amounts of cells that they're made of. And most are able to move with some form of locomotion. So legs, tails, things like that. They're able to move, which serves them well because of that heterotrophic nature. They need to find their food and capture their food. So in summary, as technology changes, classification groupings can change as well. Originally there were 2 kingdoms, but now there are 6 kingdoms within 3 domains. Okay, continuing on. Let's talk about phylogeny. So Darwin's theory of descent with modification led to the modern science of phylogeny, which is also known as cladistics. And we'll discuss how this relates to the classification in just a second. Phylogeny is the study of evolutionary history and the possible relationships between organisms. So we're taking the classification that we already discussed and we're trying to figure out how those organisms are related to each other. So not just what similarities do they have, but how might that have occurred. And these relationships are illustrated in a cladogram or a phylogenetic tree. And these represent a hypothesis of how things may have evolved based on the information and similarities between these organisms. Scientists make some assumptions about how they are related to each other. Sometimes they involve genetics in these cladograms and phylogenetic trees in order to show relationships. So here's an example of a cladogram. And you can see we have lancelet down here. We've got a leopard on this side. And these organisms become more complex as they are moving towards the right from the left to right side. And these are the ways that they are related to each other. So the lancelet is the only organism in this grouping that doesn't have a vertebral column. And so we put that after that lancelet grouping. And then the lamprey does have a vertebral column, but it doesn't have jaws. So we're going to put it here on its own branch. And continuing on here, the tuna does have jaws and a vertebral column, but it doesn't have 4 walking legs, and so on. So the leopard has all of these characteristics, and this would show that a common ancestor in this area here would have differentiated so that the lancelet would have come from that common ancestor as well as these other organisms based on their characteristics. So what are the parts of a cladogram? A cladogram is comprised of groups or taxa of organisms at the top. So those are the ones that we just showed here. These are taxa or groups of organisms at the top. The derived characters or features of an organism, like jaws, hair, etc., are all placed along the bottom of the cladogram. Generally, time progresses in a cladogram from left to right, with the more advanced groups at the end. So that would make sense here. Groups diverge from a common ancestor or node where the branches come together. So here we're talking about 2 groups of organisms that came from a common ancestor, which would be found at this node. Remember, as we're discussing evolution, that one organism does not, or one type of organism does not evolve into another, but they are coming from a common ancestor. A clade is a group comprised of an ancestor and all of its descendants. So here we have a clade, which is known as tetrapoda because they all have walking legs. Tetra, 4 poda, legs, 4 legs. And so this group here of organisms, the amphibians, and all of the organisms that come after it are known as a clade. In the same way, the reptiles and all of the groupings that come after it, which in this case just includes the mammals, are known as amniota, and this is also a clade. These 2 groupings down here would not be a clade because they do not include all of the rest of those descendants that come after the lamprey. So the lamprey and fish taxa are not a clade because all the other descendants are not included. To be a clade, amphibians, reptiles, and mammals would also need to be included because they are also descendants of the common ancestor located here. So all of these would need to be included to be a clade. So in summary, phylogeny is the study of evolutionary relationships between organisms, and it can be illustrated by a cladogram. Taxa with a common ancestor form a clade. So how can we find features of extinct ancestors? If we're making these cladograms, obviously these are sometimes based on organisms that are no longer in existence. Fossils provide evidence of common ancestors that are extinct, which means that all organisms within the species have died out. Although only a few organisms leave fossils behind, only a few types of organisms, and even only a few organisms of those types, they can provide lots of information about ancient species. And fossils are classified into a couple of different categories. They're sorted into types. Molds are an imprint in a rock that are shaped like an organism. That's probably the type of fossil you're the most familiar with. A cast is a mold or imprint filled with minerals that creates a three-dimensional model. So this would be projecting from the rock rather than indented into it. A trace is a fossil of tracks, feces, tooth marks, etc., showing behavior of an organism rather than a whole organism itself. And then preserved body parts or organisms or parts of organisms fully preserved in tree sap, tar, or ice. And we definitely have a few examples of those. How do paleontologists find the age of a fossil through relative dating? So we need to discuss the age of the fossil to determine how far back that common ancestor might have existed. And two methods can be used to determine the age of a fossil: relative dating and radiometric dating, also called radioactive dating. In relative dating, paleontologists, which are fossil scientists, determine the age of a rock or fossil relative to another rock or fossil. And index fossils can be used to give an approximate age, because an index fossil is a fossil that we already have assumed the age of. And so we can either determine, paleontologists can determine, is it younger than the index fossil? Is it older than the index fossil? But relative dating does not give an exact age. Paleontologists use the law of superposition to compare the age of fossils. And the law of superposition says that the oldest rock layers are at the bottom and the newest rock layers are at the top. So our oldest layers here in this picture would be found down here. Younger layers would be at the top. And this is because as sediment is falling on this existing rock, it makes layers above the rock that's already there. And so any fossils that were found up here in these rock layers would be assumed to be younger than any fossils that were found down here. In summary, extinct species can leave behind different types of fossils. Using relative dating and the law of superposition, paleontologists can determine the age of a fossil compared to others. So how do paleontologists then find the age of a fossil through radiometric dating? So we talked about relative dating. How is radiometric dating different? Radiometric dating, or radioactive dating, allows scientists to estimate the age of a fossil based on the amount of a certain radioactive isotope found in the fossil. And if you're following along, you can click on that link, or your teacher can. And so we're starting with an unstable parent atom, which is the definition of an isotope. It's an unstable version of an element that gradually decays over time. And if we can measure the rate that it decays over time into another form of that element, then we can figure out how much of each of these types of elements there is, and that will help us to determine how old a fossil is, because this is happening at a consistent rate. Radiometric dating requires two pieces of information: the rate at which the isotope decays, because it needs to be consistent and we need to know that already, and how much of the isotope is left in the fossil, because if we know how much is left and how quickly it decays, then that can help us determine how long it's been decaying or how old the fossil is. The rate at which half of the isotope degrades is called the isotope's half-life. So a good way to think about it is that parent atoms, which are those unstable isotopes, are decaying and creating daughter atoms or more stable versions of that isotope. And that's occurring at a consistent rate. And when both of these are equal and half of the isotope has been degraded, that's called the element's half-life. What is mass extinction? So we discussed extinction is when a species no longer exists because all of the organisms of that species have died out. And this can occur when a species doesn't adapt to a changing environment, and then it becomes extinct. This happens frequently, and that's known as background extinction because it's happening in the background pretty regularly, especially for organisms that maybe you're not as familiar with. Occasionally, however, a large event causes many species to die around the world, and that is called a mass extinction. And the one that you're the most familiar with, I'm sure, is dinosaurs. The extinction of the dinosaurs at the end of the Cretaceous period was a mass extinction event because many species died. So at what rate does evolution occur? So if we're talking about common ancestors evolving into different groups of organisms, how quickly does that occur? Fossils and the rate of mutation in DNA provide evidence showing how quickly evolution occurs, because of course it requires a mutation in the DNA in order to change an organism or population. So similarities in DNA and proteins are one way that evolutionary biologists can track the rate of evolution. And so they are looking here at different protein changes and how similar this protein is cytochrome C in a variety of different species, and comparing that to humans. So chimpanzees and humans have 0 differences in this amino acid protein chain, and then we continue on. The rhesus monkey has 1 difference. Rabbits have 9 differences compared to human, and so on. And so this comparison allows evolutionary scientists to determine how recently these organisms diverged from a common ancestor. Although Darwin suspected that evolution occurred gradually, which is a process known as gradualism, because he got influences from scientists that had looked at gradual changes in the earth and its surface, evidence actually shows that species often evolve rapidly, followed by periods of little change, which we call stasis. So this is what Darwin would have expected to happen, that these organisms are changing very slowly from one common ancestor to all of these ancestors, or all these groups of organisms at the top. And that would have been a gradual process, continual. And punctuated equilibrium, which is the newer model, suggests that there are rapid portions of time where there's rapid changes, and then there's areas called stasis where the organisms are not changing for long periods of time. So we call that pattern punctuated equilibrium. In summary, the age of fossils can be estimated through radiometric dating. Evolution does not occur gradually, as Darwin suspected, but it can be prompted by mass extinction events, which cause rapid change in surviving organisms. So these mass extinction events would drastically change the environment, and then that would be followed by a rapid change in the organisms that survived, because they are now dealing with a brand new environment.